Okay, folks, I think we'll get started. If people start straggling in, they're welcome to catch up on the stories. Today's session, it's my honor to kick off our sort of transformative innovation series here in room 206 of the Public Sector Summit. And right now we're going to be discussing sort of next generation medical analysis. And what we mean by this is how technology is enabling disruptive innovation, either within the biomedical research space or biomedical care, and how these types of technologies might change things for the better. But you know, when folks discuss disruptive innovation, they sometimes think of this, you know, a giant wave going over an industry. You have the small guys in a boat trying to survive, technology washing over them. And certainly, you know, this large wave represents a host of technologies, not just cloud, but genomics we've seen over the last 10 years really make precision oncology a reality. Cloud, obviously, enabling collaboration and at scale and doing things that were normally pretty difficult to do across institutional boundaries. We're just starting to scratch the surface of what machine learning and AI can do for healthier outcomes. As well as we saw, a really great case in public safety. So IT has definitely been in it for a while. They've been you know, taking their boat through these waves. And I think that healthcare is over here where it's about to be subsumed by these technologies in the cloud and healthcare space. But it's not like, you know, disruption doesn't have to mean destruction. Right? It can mean surviving and adapting to the waves of technology that are coming at you to make you better. And today we're going to have a couple of stories, one by Dr. Nick Weber from the National Institute of Allergy and Infectious Diseases, where he's going to be talking about the history of his organization adopting cloud models and lessons learned there. And then followed on quickly by Dr. Caleb Kennedy from the National Bone Marrow Registry, discussing about how to make better match outcomes, healthier outcomes for patients from that registry. So without further ado, Dr. Nick Weber.
Thanks, Angel. Yeah, so I'll talk about being in the boat for the past 7 or 8 years. I actually was just chatting with Angel about having been here at the Public Sector Summit every year so far, I believe, for the 7 years that it's been here. So I guess they finally acquiesced and said that I could give a talk. So I want to talk about the experience we've had at the National Institute of Allergy and Infectious Diseases, NIAID, which is one of 27 institutes at the NIH. We study flu, AIDS, Ebola, Zika, malaria, TB, those sort of diseases that are kind of associated with microbes. And I want to tell a story about how we are investigating microbes at a deeper level using AWS to kind of enable that. So as you may know, the human body has microbes kind of all over us and within us, from our head to our toes. So the head there, you see the head louse that causes lice. On the foot, you may be familiar with athlete's foot fungus, but there's a lot more changes that happen in our microbiome, which is the collective makeup of the microbes that exist in kind of a symbiotic relationship with us, that have implications on tons of diseases and afflictions, from celiac disease, Crohn's disease, arthritis, acne, obesity, type 1 diabetes, you name it, there's some sort of association with changes in your microbiome that are associated with these different afflictions. And there's a lot of interest happening over the past 10 years in kind of studying how the microbiome makeup, the genomics of these particular bugs and what they do in our bodies, how those changes impact health and disease. So you may be familiar with probiotics or heard a story about a fecal transplant to get somebody's microbiome back into a stable balance. So there's a lot of interesting research going on in this space. And we use AWS to kind of enable a lot more researchers to participate in this research. So first, I'll tell the story of how we kind of got involved in AWS, and then I'll kind of jump through 4 separate topics with a quick kind of tidbit at the end of something that I learned along the way in working in this space over the past 8 years off and on. So first, in 2009, I started as an intern at NIAID. This is what the EC2 console looked like back then. So I think there was interest by my kind of management to kind of say, everyone's talking about cloud, what can we do to learn, go off and do something. And I don't think they anticipated a whole lot. I was just out of grad school, so just tell us something at the end of a few months. So I took some essentially protein sequence data that you see on the top, and this is a lot of molecular modeling that takes a lot of heavy compute to be able to kind of put the pieces together in different configurations and then do some analysis to determine the lowest energy configuration of these molecules to determine what's the likely configuration in the human body. And then we can start to kind of do further analysis in vitro after doing this modeling. So I did some work on that for a few months using AWS. So these are actual slides that I used in 2009 at the end of my internship presentation. I won't go into too much detail, but I do want to mention that kind of I compared our internal NIH HPC cluster to AWS for the work that we did. And I found kind of two things. First, on AWS at the top, it took multiple minutes to be able to create one of these configurations, and I was creating 10,000 of them, versus on the bottom, in our supercomputer at NIH, it took just under a minute to do that. So it was less time. And it cost a lot less when I did the math out. For creating 10,000 decoys, 10,000 configurations, it would cost $70 or $80, and I was using my boss's credit card at the time because we didn't have kind of approval from NIH to do this. But trying to figure out how much does it cost? Less than $1 on our supercomputer, $70 or $80 on AWS. So it was lower cost. And the question was, why would we use cloud? But I think what I came and learned from this is that it was intriguing. There were definite benefits to this model that I hadn't ever come across before in the work that I had previously done. Even though it wasn't the right use case for us at the time, there were probably some other things that we could learn and do for which this was the right use case. So that was my lesson learned kind of back in 2009 when I first got my feet wet, is kind of focus on what you're doing and how the technologies and tools that are out there can be brought to bear appropriately. It's not a one-size-fits-all silver bullet, we all know, so kind of figure out what's going to work well for you. So moving on to 2012. So this is when the opportunity came. I had in the back of my mind, here was my experience with AWS and cloud. And NIH funded the Human Microbiome Project. So this essentially tried to find a healthy baseline of 300 volunteers to kind of figure out these people are healthy, what does their microbiome makeup look like? So there was funding done. And you can kind of see on the right here, this is the wave that Angel mentioned. So around that time in 2012, publications started to double and double. You can see the searches on Google Trends search kind of going up at the same time. So we wanted to hop on this opportunity as well as we could so we didn't get subsumed by all the kind of interest in this space that was coming. So as is kind of common in federally funded research, the government funded an organization to put the data out there. It was about 20 terabytes, and in 2012 terms, that was a lot. It's still quite a lot today. And said, here's the data, here's some tools that you can download, research community, go have at it. But that's essentially that's all we can do. And that really wasn't a sustainable model for many people who were interested in doing this research across the globe. They didn't have the storage, they didn't have the compute, they didn't have the tools to be able to do this locally. So what do we do? So that kind of prompted our problem statement at the time is, how can we work with this research community, doing this interesting and enabling research in the biomedical research space with microbiome, to be able to make this model a little bit more sustainable and not ask everyone to kind of download and use and do everything on their own. Can we bring them together in a way? So we turned to AWS. So this is a kind of screenshot of the public data that we put from the Human Microbiome Project on AWS. I think back in 2013. It's been updated a few times since then. So people were able to kind of get the data centrally on Amazon S3, hosted by AWS, and start to do some analysis on it. But that was only one part of it. So the data is there. What else? So we decided to build a tool or a platform that we call Nephele. Nephele is the Greek goddess of clouds. She uses, she wields the cloud to do her bidding. So we tried to use that in the platform that we built. So we essentially wanted to be able to answer or help researchers answer questions that they had about sequence data that they took from their patients or their subjects. So is there a pathogen? Is there something that's disease-causing found in my sample? How can I know that? Are these associated with particular clinical outcome or diagnosis or phenotype? How can I figure that out? And does my set of samples differ from this healthy baseline that I just mentioned? And what's the difference? Why is that important? So we built this tool called Nephele that enables you to kind of bring data sets, provide a little bit of information about the analysis that you want to do, and then get these outcomes that shows you the different diversity and lots of plots and outcome charts, statistics, those sort of things. So this is a technical talk, so I wanted to provide a little bit about the architecture, and I'll talk more about architecture on AWS at the end as well. So essentially, starting at the top, user will come to the kind of web front end for this tool, provide some details about, okay, here's the type of analysis that I want to do, upload their data set or point to their data set that exists out on the web somewhere, and then say go. That will trigger, going down to number 3, putting data into Amazon S3. We have a Lambda function that essentially watches that Amazon S3 bucket to make sure, okay, is there data here? Is there kind of the parameters that the user set? Once those are there, we'll spin up a new EC2 compute instance to do that analysis for users. Send SQS notifications back to the front end for the user to say, here's your job outcome, it started, you can download your data now, and kind of cycle through that. So that was our architecture at the time, but this took a number of iterations to get through. We had Elastic Beanstalk in here at one point. We had CloudFormation. We had lots of other AWS tools that we tinkered with and tried, but didn't necessarily fit what we were doing. So my second lesson learned after 2012 was that there are a ton of ways to do AWS architecture. I'm sure many of you are very familiar with that. So make it a point to try to stay up to date with the technology. The way that we do that in our team is put in a couple day spikes within our sprints to kind of say, let's look at this RDS service, compare that to what we may be doing for separate database approaches, and then report back to the team and figure out if we want to try to move in that direction. So that's 2012. Moving on to 2015, so we had implemented this Nephele platform close to the end of that period there, and we wanted to start to kind of figure out what did we learn? Is this useful for the community? So this is a chart that shows the daily analysis jobs that users were doing during a beta release at the time. So you see on the y-axis the number of jobs over time. And I think the key thing that I wanted to point out here is that if you average this out over the couple of months that we had this beta release, we would have had 12 analysis instances, jobs per user per day. And in our previous model, we could have bought enough compute capacity to accommodate those 12 on average jobs per day, or maybe a little bit more. But we'd have time when the resources were idle, and we'd have time when people couldn't get their analysis done. And these analyses take on the order of 5 hours on the short end, maybe close to a week on the long end. So if you are the 82nd person in line here, unfortunately, you're going to be waiting a couple weeks if we went with that model once you're queued up and waiting for other jobs to be done. So what was useful in our AWS model is that everyone could come at once, everyone could start their job in this workshop that we had this day, and within the amount of time that their particular analysis job took to complete, they all got to do it, got their data back within that time. So it's highly scalable, and it was a much better use case than the one I showed at the beginning with the protein configuration analysis. So what else did we learn? So we've had about 2,500 data analysis submissions. This is a kind of word cloud of what people tag their analyses with. And what's interesting is you see things like broccoli or hospital or other things like that, that we kind of had a happy accident where people were coming and saying, well, these tools can be used to do different types of analysis that aren't necessarily for human microbiome. So if I want to look at a physical environment and see what microbes may exist, some interesting research on the toilet seat handle, for example, you can take a sample there, use these tools, and be able to see what exists on your toilet seat handle. And you actually find that there's a lot of things that actually exist in the soil, and that's because so many people go and do it this way, right? So it's from their feet that the microbes get on the toilet seat handle. We also found that this number continues to grow, but in that time period we had about 500 registrations, over 150 organizations. This week alone, I think we've had 7 new countries represented of people kind of coming across the resource and signing up. And we get good analysis and tracking using tools enabled by AWS to kind of figure out who's doing what, how much is costing NIH, and how we can convince our management that this is a useful thing for us to continue doing. And then we also learned that this is a really kind of good use case for students in undergraduate and graduate courses. So here's 3 universities who used the platform that we built on AWS. The key takeaway here is that it was about $2 or $3 per job per student to do these analyses, compared to what it would have taken them to set up computing infrastructure locally, or all the things that they did before, bring in all these laptops, configure all the tools, work with their HPC community to make sure things work, all that troubleshooting and time they didn't have to do. So this really enabled them to kind of focus on what are the analysis outcomes, why is this meaningful, as opposed to what are all the technology hurdles that I have to get over to make this work. So we also have kind of a user support system that kind of automatically asks people, if the tool is beneficial, what feedback they have for us, and we kind of get voluntarily some good feedback that this is helpful and provides a resource that people couldn't do on their own because they couldn't build up that infrastructure. They didn't have bioinformatics personnel who had these capabilities, so coming to the platform was really useful to them. We also learned that over time, there's entropy in any system, so we had some challenges. So we architected, as we saw, uploading directly to S3, had some challenges with hitting some maximums and data file corruptions and moving large data sets around. We started building this, like I mentioned, in around 2013. So we have some older technologies there, a lot of technical debt within the system that we're continually trying to kind of refresh and update. And we spent a lot of time on user support. So people will say, I tried this analysis, it didn't work, can you help me? And we're kind of going back and trying to provide workarounds as we're building some new pieces to the component. So support is important, but I think where this ultimately led us is we came to a decision of, should we continue to patch and revise and make incremental increases to the system, or should we rebuild it a few years later? So if you remember the title of my talk, Building and Rebuilding, you probably know the answer. But we considered providing updates incrementally to users, or saving time by automating and doing deployment, saving time on operational costs by using some other services and features that will make the system more efficient, and then turn this into a more generic platform that could be provided to researchers who are not only doing microbiome genomic data analysis, but any sort of data analysis that could be standardized in a Docker container or in some sort of VM that a scientific researcher might want to do. So takeaway here, evaluate continuously. So make sure you're keeping metrics and understanding who your users are, and be willing to tear down and rebuild if needed. I think that's something that we kind of had a hard time grappling with, but ultimately decided that's the right approach for us. All right, finally, 2017. So here we are, present day. I'm going to talk about where we're going. So this is my second architecture slide. I'm going to build through this one as compared to the previous one. But essentially, this is the kind of new version of Nephele that we're rebuilding presently. So you see time 0 at the left-hand side, so we have multiple states that I'll walk through. But essentially, a user comes, and instead of uploading to S3, they're going to upload directly to the network file system, the EFS file system on AWS. They'll select their job parameters, just like they have before. I want to do this sort of analysis using these tools and cutoff values and things like that, and then they'll submit their job. So the user does those 2 things, and then the system kind of takes over. So we'll send the job parameters to the RDS database service. We will drop a message into a queue for pending jobs to say there's a new job coming, and we're ready to process that. So at time 1, once all of that happens, we set up a new queue that's tied directly to an EC2 environment that's set up to do this particular data analysis. We've attached the file system, the same EFS file system, to that EC2 environment, and we have kind of this central, ready-made place to start to pass messages through to do the analysis that we have. So now, a couple minutes later, the EC2 environment spun up. I have access to data. I'm ready to go in this ephemeral environment, so we can start to pass those messages in. We query RDS to get the information for the job. We say, start this job. We write the output back to EFS and indicators back to our RDS database service, so we can query to say this job passed, this job failed, it's at this step, whatever. And then ultimately, when the job completes, we send results to Amazon S3. We use SES to notify the user that the job has completed or if it failed out. And we pass a message into that same queue to say, shut down this ephemeral environment. That's done. So then, as you see, we can return back time point 1. And just like the current version of Nephele, this is scalable, so if you have lots of users coming in and doing this in a workshop, 100 people coming at once, it just spins up a bunch of different resources and shuts them all down, as opposed to queuing anything up. So I think this architecture is going to be better for what we're doing, more cost-effective, and much easier to automate into, which is what I'll kind of talk about and close in. So what are we doing beyond our particular single Nephele microbiome analysis system? So a different team within NIAID is building a platform as a service that we're dubbing Monarch that has lots of these open source components tied to AWS, but also established in our internal infrastructure with the ultimate goal of saying, I have a new application or a new service or a new tool or utility. I can run that internally, or I can run that in the cloud, and it doesn't really matter. That's a business decision and not a technical decision. So we have all of the same kind of services and automation happening internally that we do working on the cloud. So you kind of compile your source, build into a Docker container, have automated security scanning, lots of other components that build out your environment. This is kind of the DevOps concept that you probably heard in some of these other talks. And we run all of these things on AWS in the Monarch platform, log to a central logging service that also logs information from internal servers and systems, and have lots of monitoring and other components in this space. So the kind of core concept behind here is that we have an environment set up. We can deploy new applications to that in an automated fashion. So if I am a developer, I commit my code, I can automatically kind of build out that application in a container, in an environment that is all fully managed within this Monarch platform, within VPCs, and has access to all of the services and accounts that it needs. So kind of the core concept here is that doing this in a simple way with some automation tied to it and security established alongside it with UC Datadog and log entries, but also CloudWatch and CloudTrail, the AWS services, we can get kind of a stable solution that we can continue to build on and that we think application developers and the rest of the research community at NIAID is going to be able to kind of grow into. So my last takeaway then is take the time to simplify what you're doing, automate as much as possible, focus on security. We heard that yesterday in the kind of keynote, and look to create or adopt or buy a platform that can scale. I think that's really going to go a long way in lots of your kind of use cases. So to close, here are my 4 use cases, but or 4 lessons. The key thing I want to mention is, it's not important to kind of just do the technology for the technology. You have to kind of have some interesting, challenging, valuable use case like we have over here. Get your good people working on these kind of hard problems, using these cool new technologies to solve those problems in whatever space that you're in, and I think you'll have good outcomes. So thanks for the time. A bunch of acknowledgments here at the bottom, a lot of people who've participated in this work, but I'll turn it over to my colleague, Dr. Caleb Kennedy from Be The Match, and we may have time for questions at the end, or we may not.
Thanks, Nick. So I'm going to just keep going. I wanted to talk a little bit about who we are as an organization. So I'm going to briefly just describe. Many of you probably don't know who we are unless you've been the recipient of a bone marrow transplant or have donated to one. So we're a nonprofit organization. We've been around for about 30 years. We're based in Minneapolis, Minnesota. We have about 950 or so employees. And we're probably best known for operating the largest and most diverse member registry in the world. So these are people who would ostensibly give bone marrow to a patient in need. These patients are typically suffering from severe blood disorders, leukemia, lymphomas, and some other inherited diseases. So what do we do? We do a lot of things. We don't actually facilitate bone marrow transplants. We operate through a global network of partners that include transplant centers, laboratories that do the genetic testing. In those terms, the most important factor for a match with good outcome is the human leukocyte antigen. So it's a system of genes that determine self from non-self. So your immune system is constantly scavenging cells in your body and looking for those that have altered themselves or are infected with viruses. And it has mechanisms, lots of biological mechanisms to eradicate those cells. So that can arise in a lot of different contexts. Cancer is one of them. So this is just a graphic to display the most important, it's not the only important factor, but it is the most important factor, is that genetic matching between donor and recipient. We also do a lot of work on the back end. So post-transplant, we're very interested in the efficacy of matching and improving matching, both with HLA as an important genetic system, but also genome-wide. So there's a lot of information that's not captured in that small system of genes that does affect both positive and negative outcomes for transplantation. And so just to give you a clue, this is a graphic that describes many of them. HLA is part of the major histocompatibility complex, and there are minor histocompatibility antigens as well. That's basically everything that's not HLA. And they form the basis for the positive, what we call graft versus tumor or graft versus leukemia effect. That's the beneficial part of matching and transplantation. But if you think about it, what you're actually doing is transplanting an entire immune system from one donor into a recipient. And if you don't get the matching right, you can have the effect of what's called graft versus host disease. So it's a systematic effect where the donated immune system sees everything as non-self and attacks it. And it's a devastating disease. It happens often. Up to 70% of transplants have some form of acute or chronic graft versus host disease. So it's a challenge, and it's one that we're dedicated to solving. One tool, obviously, that we have, and a lot of others, is the rise of genomics and the garden of technologies that have developed kind of contemporaneously. Probably the most talked about is next-generation DNA sequencing. So that enables us to get large amounts of biomolecular data from donors and recipients, from individual cells within transplantation, at a very low cost and an ever-dropping cost. So this is a graphic that makes it into almost every genomics talk. And alongside the cost per genome curve, you see Moore's law, which is the compute capacity on a single CPU. What's not really discussed too much, and I think where AWS and cloud computing technologies in general are enabling, is the distributed compute that a lot of people are taking advantage of. So the curves would probably look a lot similar, or they'd be closer together if we were talking about distributed compute capacity, which we will in just a second. Nevertheless, there's this glut of data and an ever-increasing amount, which makes the interpretation of those genomic data paramount. In our context, what I'm showing here is a graphic that one of my scientists put together, which I thought was one of the most beautiful figures I've ever seen, but I had absolutely no idea what it meant when he gave it to me in an email. So he added some annotation, and I still didn't understand it. What it does is shows the genetic variance along the outside of the curve, genome-wide, so this is chromosome by chromosome. The outcomes in terms of graft versus host disease in this case, in the middle concentric spheres, and then you can actually connect genes that are related to each other in functional networks interior to that. This is all just to say that while we can get a lot of genomic data and analyze it fairly quickly, interpretation is still a major challenge for us. And AWS is really helping us to do that. We've taken this and kind of parsed it out, and we're learning some really interesting things about minor histocompatibility antigens, specifically in male patients with female donors, because they have a Y chromosome that is inherently not self, that can affect outcomes negatively in that case. So how do we do it? I'm going to go through some use cases, just a few. We do it in a lot of ways. Everything from the front end registry, so matching donors with patients in need, all the way back to the interpretive outcomes research that I just described. So we started using Amazon pretty early on, about 3 years ago when I first joined the organization, to do rapid prototyping and development. That really was the motivation for it. Rather than invest a bunch of money in IT infrastructure, and an internal on-premises compute that we didn't really know we needed until we proved it out. We decided to do a lot of that on the cloud. We operate, like I said, through a partnership of a lot of laboratories that do the genetic testing on our behalf, and we add about 2,000 or so members to the registry a day. So it's not big data by most standards. HLA is a small system, but we add a lot of members, and the velocity of data is quite high. So what we came up with was a public standard for structuring these data and metadata called histocompatibility markup language, which is a terrible name. But it's an XML-based standard that we give to our labs. They put the genetic data from donors in that standard, and then they submit to us through interfaces that we've all deployed on the cloud. We've used a lot of internal services of AWS, again, not things that we wanted to develop in-house from scratch, for load balancing, for containerization, for different development and production environments, for validation of the data before they enter the registry, because obviously once they're matched to a patient, you don't want to go back and say, well, the HLA was wrong. That would be devastating for the patient on that side of things. And then we still, once the validation has happened, we still pull the data on premises through a firewall. Mostly for security reasons. We're regulated by a lot of agencies. The FDA is one of them. And we've moved slowly, I think, with those agencies. Although they're very receptive to cloud as an enabling technology, they definitely want you to hold hands with them and walk the path so that they learn. One major benefit of this is flexibility. So as we learn more from the outcomes-based side about what is important for matching, we want to add it as quickly as we can to the genetic testing component of donors, which is cheap and easy, like we said, but can be a glut for our information systems if they can't react to it very quickly. So HML is flexible in and of itself, and we wanted that flexibility to be represented in our entire technology stack, which AWS has enabled us to do. This is the actual matching portion of it. I'm not going to talk too much about it. Mostly, there's some descriptive technologies that we're using. A lot of this is centered around distribution. So we want to do as many searches as we can, as fast as we can. For every search that is what we call productive, and we do about 6,000 productive transplants a year, there are many searches. There are probably tens to hundreds of searches that actually go on for every match that is found, where people, transplant centers or physicians are customizing the search depending on their patient's specific needs. So the 6,000 transplants we do aren't really representative of the flux we need and velocity of searching and matching. We also capture all of the searches in what we call Search Archive. And though we haven't implemented a lot of AI to search this, we're starting along that path to deliver better search results to physicians faster. Most of this is enabled on Spark architecture, which has come along in the last couple years. Hadoop had definitely a technology and cognitive barrier, but Spark seems to be much easier for our developers to capture and start developing very quickly. Then finally, I want to talk a little more on the outcome side of things. So we're increasingly doing genomic analysis on our donor-recipient pairs. We have a biorepository of all of our donor-recipient pairs. We capture both the recipient tissue, mostly blood products, and the recipient, and they're then accessible for genomic analysis. So very recently, in the figure I showed before, we've engaged in a partnership with Human Longevity to sequence 250 donor-recipient pairs. That's 500 samples total, half with GVHD and half without, to try to understand what outside of the major histocompatibility complex is causing the disease in that case, because these are all 10 of 10 matched donor-recipient pairs. That doesn't explain it. So in order to enable that, there's a lot of need. There's data storage and data access and data transfer. We went to our IT department and asked for 100 terabytes of storage access through our service center, which is our ticketing agent, and there wasn't an emoticon to capture their response to that particular request. There actually probably is, but it's inappropriate. Below the Spark line is, and including the Spark line, is pretty much agnostic technology that you can point at any direction. But what's really fascinating and is increasingly enabling is what's above the line. So a lot of specialized expertise that's being used to develop genomics-focused applications on top of all this enabling technology. So you no longer have to do, a lot of distributed compute batch processing, which can be very difficult in a genomic context where the interactions between data are just as important, and it's not always easy to figure out how to parallelize that. But Spark is sort of natively parallelizable, and if you can get your data to live on that ecosystem, you can start to add in all of the interpretive tools that are critical for specific genomics and clinical research. I'm not going to go through any more detail than that, except to say that now we have much of this infrastructure, and we're starting to build on top of it a lot of the artificial intelligence and machine learning approaches that naturally lend themselves to a complex problem like the one we're trying to solve, where you have definitive outcomes that you can use for training and testing, and you have large integrated multifactorial combinatorial effects going on in your biomolecular data. So that's a direction we're pursuing at this point as we sort of wrap up this first cohort analysis. And then just a little bit of discussion about where we think we're going. Bone marrow transplant has been around for a long time. It's curative for leukemias and other blood disorders. But actually, if it weren't for the negative effects of the preconditioning regimens that patients have to go through oftentimes, and graft versus host disease, it actually has a lot more application, clinical application, outside of those contexts. Things like autoimmune disease, solid tumors, cancer immunotherapies, which are a big area of focus and research right now in the healthcare space, other heritable disorders, and then even things that aren't thought of usually in the same breath as bone marrow transplant, which is solid organ transplant and even pregnancy. So like I said, we're working on identifying individual minor histocompatibilities that are on the Y chromosome and reactive or immunoreactive to female donors. But if you think about it, pregnancy is almost exactly that. So it's if you want to think about a male donor and a female recipient, and though it's not as adorable as a little baby, the fetus is a haploidentical allograft. That's the technical term. There is an immune response to that sort of hybrid mosaic tissue in a female, and it can lead to negative outcomes like preeclampsia and miscarriage in some cases. So while we may never actually get in that space, I think the things we learn from the model system that is a bone marrow transplant will be enabling and informative in a lot of different clinical spaces. So with that, I'm going to complete, and I think we may have a couple minutes of time for some questions. Thanks.
All right, that was a great set of couple of talks. Thank you both. We do have time for one or two questions. If anybody has it, just raise your hand and in the back of the room there, just yell and I'll repeat.
Cluster architecture, there had to be specific reasons you went that route.
Yeah, I think
The question just
sure. The question is, what are the specific advantages of Spark over Apache Hadoop? And so in our group, I'll be honest and say we enable the developers to choose technologies. And as long as they can provide us with validation and sort of rationale for why they've done it, that's all we really require. And developers are almost always on to the next thing. So it's a lot of times very difficult to anchor them to a particular technology or particular way of doing things. They want to they want to continue to innovate. They want access to the newest and best and brightest tools. We talk a little bit about it as butterfly catching, which it is. And as long as they can bring back to us that it costs about the same, it goes about it takes about the same amount of time, and on our validation sets, which we have extensively, it gives the same results, or they can justify differences that are scientifically valid. So I think that's one big reason, is we give the developers broad latitude to choose technologies. The second has been, it's related to that. I've found, so I did a bit of Hadoop analysis and coding in the day. It seems like the ecosystem of tools, scripting tools and otherwise, just make MapReduce and the whole paradigm a little more accessible with Spark. I found that to be true with the technical and non-technical folks. So we have dedicated engineers, but we also have a large number of scientists who are programmers, but they're not computer scientists. And I think that that's probably another big reason. I can't point to a specific technical reason. They're mostly cultural.
Okay, thank you.
Go ahead. Yep.
I am David Michael, formerly QIAGEN, and we did the HPV testing diagnostic before transplants. And with the microbiome and with what Nick presented, do you see that being a potential future gauge? And are you tracking the success of introducing genomics to transplants and the success of acceptance from donor patients? And what's that trend looking like?
Yeah, that's a good question. So we were in a strategic meeting with some of our board members and executives, and this exact topic came up. I think enabled a lot by the genomics technologies that are in place for doing metagenomics. And the question arose, well, should we take a role in effectively poop transplants? And so we were talking about poop transplants with our board members and a lot of executives. And if that's not disruptive, I'm not sure what is. There is an emoji for that one, actually. And so, yeah, we are thinking about it, because what we're learning is that, again, back to the last slide, there's a lot of interaction between the immune system and all of the other things the body does. It's critical for pretty much every physiological system. And so we use science and technology to just sort of follow that path. You know, it's not predetermined. Research is going to lead the way as to how effective on an outcomes setting that is for bone marrow transplant and other stem cell transplants, but it's definitely something we're thinking about pursuing. Nick, I don't know.
Say anything? Last word.
On this topic, I think I mentioned it, right? So microbiome and metagenomics has been exploding for the past 6, 8, 10 years, and I think there's tons that we can learn. And if we can build kind of enabling platforms and technologies to make it easier for people to do this sort of research who couldn't do it before, I think that's the best way to get there. So that's kind of been our goal all along, is to kind of enable or democratize access to people who didn't have the local infrastructure, local expertise, build something that they can just come with their data, bring your own data, do your analysis, get some results, and kind of move on from there without having to worry about the technological setup, abstract that away. I think that's how we're going to kind of, from our space, enable the community to find new discoveries in this area and plenty of others. So it's an exciting time, and I'm happy to chat with others after we're done about any of this. So thank you.
We'll be available here and just outside the room. With that, I'd like to thank our speakers and get you guys into your next session. Thank you all for coming.